All right, advancements in 3D bioprinting with computational models for enhanced cell viability and functionality.
Okay, thank you.
Forward, bottom one moves backwards.
So I said it one more time.
Big one moves forward, bottom one moves backwards, and hold that down for the portrait.
Okay, hopefully I just need to go forward. It's too complicated for me. Okay, good morning, everyone. I'm going to talk about a couple of projects. I only realized I only have 15 minutes, so I will go a bit fast. But that'll give us some general idea that we can discuss it at the break you know, in the breaks or you know, in the afternoon sessions. So these two projects, as I mentioned, are related to 3D printing. There are basically, I want to say that in 3D printing, as you know, the 2D culture has, you know, their own issues, and they don't reflect really the actual, you know, tumor inside the patient. So people have started to look into, you know, 3D or spheroid, you know, culture of cells, and you know, more recently there has been the many research going on about, you know, using 3D bioprinting for, you know, for growing cells, and then putting them on a chip for, you know, patient on a chip or tumor on a chip. And that has been the direction in my lab over the last few years. So in bioprinting, there are usually two cases. One is, you know, this what we call two-step bioprinting or one-step bioprinting. Two-step bioprinting, usually you have a scaffold that is pre-made, and then you start adding cells to that, and then you grow them. And in one-step bioprinting, you co-culture them all at the same time. So you add cells, you know, biomaterial, everything you need in the nozzles, and then you print them at the same time, as you can see in that picture. But it has its own challenges. One challenge is the biomaterial selection. There are so variety of biomaterial that we can use, different concentration, different crosslinks, and so on. And also, we don't know what's going to happen with the cell viability after we culture them, we do the 3D bioprinting. Are the cells alive? Are they going to survive or no? And finally, what happens if we culture them for, let's say, for 10 days? What we're going to observe, given the structure of the cells, what's going to happen to these cells after 10 days, so we can start to do treatment and, you know, do sensitivity for drug sensitivity for the cells. The idea, as I mentioned, is that, you know, we take, you know, samples from patients, we grow them this way in the lab, and then, you know, try different drugs and see which one's going to work for the given patient. So that's the ultimate goal. So usually in the experimental thing, we just do trial and error, and we just, you know, run for different experiments for different, you know, concentration, different, you know, time, pressure. So the bioprinter has its own, you know, characteristic, temperature, pressure, nozzle size, many variables over there that you can choose. But the idea would be to apply, you know, machine learning techniques, as well as, you know, mechanistic modeling, like, you know, simulations, ODEPs, and things like that, in order to try to optimize this, you know, bioprinting system. And there are two, you know, optimization problems for us. One is during printing, how we can, you know, design the parameters such that we get the maximum, you know, cell viability, and also after bioprinting, you know, how we can use, like, let's say, like a cell automated simulation in our case, to study post-printing behavior of population of cells, you know, embedded in this 3D bioprinted structure. So the first project was published in Biofabrication this year, and you know, the driving force of this project was my PhD student Dorsal Mohammad, you know, Rezai, and in the second project was in collaboration with, you know, Jibin and his PhD student Sarah, which is now I just happen to know that she is now an assistant professor in Sweden. So this is the bioprinting that we do in the lab. So this is my lab here that in Waterloo that we are doing this. As I mentioned, there are different factors. So if you think about the bio-ink, you know, formulation by itself, cell type could be different, kind of it, and the concentration of biomaterial, how many cells you start with. And then with the 3D bioprinting setting, there's temperature, you know, bed temperature, nozzle size, you know, pressure, speed, and so on. And also a cross-linking, you know, amount of it, time of it, and, you know, the time, you know, of cell viability measurement. These are all different factors that are going to affect your outcome of the 3D bioprinting. And as I mentioned, the question is, how do we do the optimization problem? So the objective was, in this first project to, first of all, like any other, machine learning algorithm, you need as many as data as possible. So trying to collect as many data as possible. And then we both develop both classification and regression algorithm to predict cell viability, and then, do optimization and see for which given parameter you have the highest, cell viability. So this is the overall, picture that what we have done. So the biomaterial is gelatin and alginate, and then we use MDA MB231 cell. These are the triple negative, cancer cell, breast cancer cell lines, which are, one of the most aggressive ones. And so we, at the same time, we put them in this in a nozzle. Cross-linking will help us, it's like CL2 we add to the solution, and that helps to make the gels that we need for the structure. And then we do the printing process. As I said, these are all the parameters that can change, and eventually we're going to do a live depth assays and use confocal microscopy at the same time to look at the cell viability. So we also looked at the literature for, available data, so that because we collected, I had a couple of students over the summer, undergraduate student who did this experiment. We collected over close to 200 data sets. And also we look at the literature. We found, 500-600 data sets in the literature as well for the same type of printers. And then we collected them all in this table. And then we did, as I mentioned, this is, for example, an example that what we are going to observe in the experiment. We know, for example, the concentration that we have used for the biomaterial. This gives me, for example, 57% viability. This guy gives me 68%. Higher cell viability, the better for us. And then we did this, classification and regression problem. I'm going to go a little bit fast here because there is not much time. And then for both cases, when we want to do classification, we just divide to, higher 70% is good, less than 70% bad, and then the regression problem. So for both test and, for training and test data, did an okay job, like, all close to, 80%, accuracy in the model. And then we use the Bayesian optimization. This is an optimization problem that we could use, try to say that for which inputs, then we can get the maximum, viability that we are interested in. And the neural network was like a black box inside our, optimization problem. So basically, instead of going back to the experiment and do the experiment, we use the neural network to say, okay, given this, parameters from the Bayesian optimization, what is the outcome? So this is assuming that the neural network does a great job, which is not true. It's just up to 80% accuracy. So keep that in mind. And the result, again, was okay. So we tried to come up with we designed these two experiments. We wanted to validate. So we designed two experiments based on our, optimization problem. So these are the parameters that, the system gave us, the Bayesian optimization gave us. We call it experiment 1 and experiment 2. And then we went to experiment, and then we did the experiment one more time. So this is true validation, no cheating here. And then we realized that we were able in experiment 1 to increase the cell viability to 85%, not something that we've never got in any of the experiments. So that was a big achievement, that we were able to, increase the cell viability. And then the next one, so this is again kind of a conclusion of what we did there. So I'm going to go quickly to the second project. That now that we have the high cell viability right after printing, how we can predict what's going to happen, let's say 10 days, from the printing. And that's the collaboration that I mentioned with Jimin's group and his PhD student Sarah. And then in this work, the idea was that, can we use a cellular automated simulation? And given the structure that now we have and some of the measurements that we can do for the cell properties, how I can predict the cell behavior after, let's say, 10 days or 11 days after the experiment. So again, we designed the experiment first. So we printed cells with, again, the same scenarios, so exactly the same as before. We did this by printing, and then we did the cross-linking. We look at the cell viability, confocal microscopy to look at the number of, live cells. And then we did the cellular automated simulation with for the same structure, and we wanted to compare the two together. So that was the idea. So we use the MTT assay for, which shows us, rapid proliferation in the first 7 days, as you can see here, and then, slow down from 7 to 11 days. The question was whether cells are died and after that, or just they don't proliferate. To answer that question, we look at another biomarker called Ki67, which is a biomarker that shows us whether cells are in active phase or no, they're in quiescent phase. And then it showed us actually from 7 to 11 days, probably due to the lack of, space or, nutrition, they go to this, inactive phase, like G0 phase. And so cells are alive, but they just don't proliferate. And then we started thinking about this in silico study to see how we can compare them. So that's the main structure of the comparison between the in vitro and then our simulations. Just a little bit of details there. And then just like any other cellular automated simulation, we start to provide the rules that we have, how the cells are going to proliferate, whether we have cell movement or not, cell death. So we applied all these rules. And then this is a comparison between the simulation and again in vitro results. And you can see the simulation on the right-hand side that how these cells grow. That was the initial structure that was given to cellular automated simulation. And the number of proliferative cells after measurements and in silico. Again, there is no fitting here. We just use the experimental data trying to do the simulation and was really in a good comparison. And here are some again from the simulation. You can see that it's kind of close to what we observe experimentally as well. So overall, I guess these two projects show that using this integration of machine learning and mathematical model and simulation, we can provide a good model that now we have in the lab. This is kind of systematic in the lab that now we can, if we want to design any new experiment, we are using our optimization algorithm to design the parameters for the given cells. And then we use the cellular automated simulation to kind of get an idea about what should be the structure of the printing cells. And then based on that, any experiment that we do use now in the lab, we run this in algorithm. It's not perfect, of course, but it does really, comparing to the lab, before that we had to do hundreds of experiments, try and error. Now we are now limited to just a few number of experiments to get the highest cell viability. And in the future direction that now we are doing is that we are now doing the co-culture cells. We are doing the same experiment when we have the immune cells co-culture with the actual cancer cells, and we want to combine. And also, as I said, we also looking into the effect of treatment like chemotherapy and then immunotherapy for this sort of cell cultures, 3D cell culture. Okay, thank you so much.